Okay, picture this. Right now, at this exact moment, there's a high-stakes, completely invisible war happening all around us. In fact, it's happening inside our very own bodies. It's a relentless battle between human medical innovation and literally trillions of microscopic, constantly evolving pathogens. Welcome to this explainer on the science of antimicrobials. Today, we're going to explore the incredible weapons we've built to fight off infectious diseases, how they actually work on a molecular level, and, frankly, the terrifying ways that these microbes are learning to fight back.
So here's a quick roadmap of what we're covering today. First, the dawn of antimicrobials. Second, the mechanisms of microbial attack. Third, testing antimicrobial strength. And fourth, the threat of resistance.
All right, let's kick things off with Section 1, the dawn of antimicrobials, from deadly infections to miraculous cures. You know, it's wild to think about, but before these breakthroughs, something as simple as a scratch from a rose bush or just a common bug could easily be a death sentence. Humanity was pretty much defenseless. But then the early 20th century hit, and things changed fast with this exciting wave of accidental, serendipitous discoveries. In 1904, Paul Ehrlich figured out that a dye called trypin red actually worked against sleeping sickness. Then jump to 1927, Gerhard Domack finds Prontosil red, which your body amazingly converts into a sulfur drug to knock out staph and strep. But the real game changer? 1928. Alexander Fleming discovers penicillin, naturally produced by a fungus. And by 1944, Selman Waxman discovered streptomycin, literally just by screening microbes he found in everyday dirt. In just a few decades, we finally had our own microbial weapons. Now, as this new medical arsenal grew, scientists quickly realized these drugs had different ranges of effectiveness, what we call their spectrum of activity. Basically, it's the difference between using a highly trained sniper versus throwing a massive wide net. A narrow-spectrum drug is your sniper. For instance, aminoglycosides specifically target gram-negative bacilli. Those are rod-shaped bacteria, while completely ignoring innocent bystanders. On the flip side, a broad-spectrum drug is your wide net. It takes out a huge variety of species all at once. Super powerful, right? But it definitely comes with collateral damage, because it can wipe out your body's normal, healthy bacteria in the process, which can sometimes lead to nasty superinfections. Moving right along to Section 2, mechanisms of microbial attack. Basically, how our weapons actually work. So how exactly do these drugs assassinate pathogens without, harming the human who's taking them? It all comes down to selective toxicity. Our drugs are designed to target structures or processes that the microbe absolutely needs to survive, but that human cells either don't have at all or do totally differently. On this molecular battlefield, there are 5 primary strike zones. Our drugs can inhibit cell wall synthesis, they can disrupt the cell membrane, they can block the bacteria from building proteins, inhibit their nucleic acid synthesis, which means messing with their DNA or RNA, or they can act as anti-metabolites, basically sabotaging the bug's vital chemical pathways. And we can visually map out exactly where these different classes of drugs strike inside the bacterial architecture. It's like a giant puzzle. Down in one area, you've got drugs targeting the cell wall, which is made of this stuff called peptidoglycan, a material humans don't even produce. Over in another section, drugs like macrolides or tetracyclines are attaching themselves to bacterial ribosomes to completely jam up their protein factories. And then you have drugs that specifically stop the delicate unzipping and copying of the microbial DNA. It is a highly coordinated, multi-front assault. The aftermath of that assault, it's absolutely devastating for the bacteria. When a cell wall synthesis inhibitor like penicillin goes to work, it blocks the structural cross-linking in the bacterial cell wall. Without that rigid physical support, the massive internal pressure of the cell is just way too much to handle. The bacterium essentially blows out. It lyses, literally bursting apart entirely. And again, because human cells don't have that specific peptidoglycan wall, the antibiotic just floats right past our cells without causing them to pop. Let's move to Section 3, testing antimicrobial strength. Time to measure the firepower. Taking all this from theoretical warfare into an actual clinical laboratory, we have to ask, how do we measure if a drug is actually working? Well, the magic metric here is called MIC, or minimum inhibitory concentration. Yes, it's a bit of lab jargon, but it's absolutely crucial. It's the absolute lowest concentration of an antimicrobial agent required to stop the visible growth of a specific bacterium. Finding that precise MIC is super important because you want to use the exact dose needed to neutralize the pathogen without pumping a patient full of unnecessary medication. One of the classic ways we figure this out is the Kirby-Bauer disk diffusion test. You basically take an agar plate, covered in bacteria, and drop in these little white disks that are soaked in different antibiotics. As the drug slowly diffuses out into the jelly-like agar, it kills the bacteria around it, creating these clear halos. We call those clear areas the zone of inhibition. Generally, the larger the clear zone, the weaker the bacteria is against that drug. But if you see the bacteria growing happily right up to the very edge of the disk, yeah, that tells the lab that the microbe is completely resistant to that antibiotic. Another really precise, quantitative method is the broth dilution test. Imagine a lineup of test tubes. They all start with the exact same amount of bacteria, but as you move down the line, the concentration of the antibiotic gets stronger and stronger. If the tubes look cloudy, that means the bacteria survived and are multiplying. But right at the point where the tubes suddenly become crystal clear, that exact transition, the very lowest concentration where the liquid stays perfectly clear, boom, that is our exact minimum inhibitory concentration. And scientists took these concepts and built something even cooler, the E-test. This is such an elegant method because it brilliantly mashes up with the diffusion and dilution techniques. Instead of a little round disc, they lay down a plastic strip that has a gradient of the antibiotic built right into it. As the drug diffuses, it creates this really distinct, beautiful teardrop-shaped clear zone. To find the exact MIC, a technician literally just looks at where the edge of that clear teardrop crosses the numbers printed directly on the strip. It's highly accurate and incredibly easy to read at a glance.
And that brings us to Section 4, the threat of resistance. The microbes strike back. Here's the scary part. Microbes reproduce incredibly fast, and they are constantly evolving sophisticated new shields to survive our best drugs. There are 4 major ways they pull this off. First, drug inactivation. Some microbes actually produce enzymes, like Penicillinase, that literally hunt down and destroy the antibiotic molecule before it can even work. Second, decreased permeability. They just change their outer receptors, so the drug gets locked out of the cell entirely. Third, drug pumps. They build specialized proteins in their membrane that act like nightclub bouncers, constantly grabbing the antibiotic and spitting it right back out. And fourth, altered binding sites. They mutate their own internal targets, like their ribosomes, so the drug's key just doesn't fit the lock anymore. And I want to be clear, this microscopic science has terrifying real-world consequences. When bacteria acquire these specific resistance mechanisms, they level up and join what we call the superbug roster. You've probably seen some of these on the news. MDR-TB, which is multi-drug-resistant tuberculosis. MRSA, a nasty staph infection that just completely ignores standard antibiotics. VRE, and Carbapenem-resistant Enterobacteriaceae. These are serious pathogens that are increasingly just shrugging off our standard go-to therapies. It makes previously simple, treatable infections incredibly difficult, and sometimes downright impossible to cure. But hey, human innovation isn't exactly backing down either. One of our coolest laboratory counterattacks is a phenomenon known as synergism. When we place 2 specific antibiotic discs near each other, like combining amoxicillin clavulanic acid with aztreonam, their overlapping fields create a massively amplified clearing zone. It's a synergistic effect that is way more powerful than either drug could ever be on its own. By pairing treatments up like this, we can sometimes bypass those microbial defenses entirely, basically outsmarting the superbug at its own game. Which really just leaves us with one massive hovering question. We've seen the brilliant scientific discoveries of the past century and the incredible molecular weapons we wield today. But as these microbes continually mutate to survive, building new shields and stronger pumps, will human innovation outpace microbial evolution in this endless arms race? Because the science of antimicrobials definitely isn't a closed book. It's an active, urgent, and constantly shifting frontier of discovery. Keep questioning, keep learning, and thank you so much for joining me on this explainer.